Hello students, this is Mrs. Sonal Patil. We had begin with the chapter Human Reproduction, in which we have studied about male reproductive system and female reproductive system. Today we will begin with the next part of the chapter. Firstly, we have already studied about testes in the last class, but transverse section of the testes we will study once again with the diagram. Each testis is an ovoid body whose structural and functional unit are the seminiferous tubules. So in the last class we have seen that testes are the male gonads. When the transverse section is taken through the testis, seminiferous tubules are visible, and these seminiferous tubules are nothing but they are the structural and functional unit of the testis. In a cross section, they appear rounded or oval and are held together by means of connective tissue. The walls of seminiferous tubules are lined by germinal epithelium. Each seminiferous tubule is lined by two types of cells: male germ cells, that is spermatogonia, and Sertoli cells. So there are male germ cells and there are Sertoli cells. When the section is taken through the testis, these two types of cells are visible. The male germ cells will undergo meiotic divisions, finally leading to sperm formation. So you can see here there are spermatogonium and in between there are Sertoli cells. Now these spermatogonium undergo meiotic divisions and finally they form the sperms. While Sertoli cells provide nutrition to the germ cells. So Sertoli cells are providing nutrition to the germ cells. The regions outside the seminiferous tubules called interstitial spaces contain small blood vessels and interstitial cells or Leydig cells. So in the interstitial space there are presence of Leydig cells. Very, very important question. From exam point of view, there are presence of Leydig cells in the interstitial space. These Leydig cells synthesize and secrete testicular hormones called androgens. So Leydig cells will secrete the testicular hormone, that is androgen. So this is about the TS of testis, which is visible when the transverse section of the testis has been taken. This particular diagram is observed. So testis is made up of testicular lobules which are having 1 to 3 seminiferous tubules, and these seminiferous tubules are lined by two types of cells called as male germ cells and Sertoli cells. Coming to gametogenesis. What is this gametogenesis? Gametogenesis. Genesis is the process of formation of gametes. So here gametogenesis is the process of formation of gametes. Let us study about how the male gametes and the female gametes are formed. The primary sex organs, the testis in males and the ovaries in females, produce gametes, that is sperms and ovum respectively, by the process called gametogenesis. In testis, the immature male germ cells spermatogonia produce sperms by spermatogenesis that begins at puberty. So at puberty, the male germ cells which are present in the testis, they will undergo division to produce sperm, that is they are undergoing the process of spermatogenesis. The spermatogonia present on the inside wall of seminiferous tubules multiply by mitotic division and increase in number. It will divide and increase its number. Each spermatogonium is diploid and contain 46 chromosomes. Some of the spermatogonia called primary spermatocytes. You can see here there are primary spermatocytes periodically undergo meiosis. A primary spermatocyte completes the first meiotic division, leading to the formation of two equal haploid cells called secondary spermatocyte, which have only 23 chromosomes each. So primary spermatocytes are giving rise to the secondary spermatocytes which have 23 chromosomes. Once again I will repeat this. Each spermatogonium is diploid and contains 46 chromosomes. Some of the spermatogonia called as primary spermatocytes periodically undergo meiosis. So primary spermatocytes are undergoing meiosis, and these primary spermatocytes complete the first meiotic division and they lead to the formation of two equal haploid cells called secondary spermatocytes. Haploid means what? Their number of chromosomes are reduced to half, which have only 23 chromosomes each. Now the secondary spermatocytes undergo the second meiotic division to produce four equal haploid spermatids. Remember, meiosis, meiotic 2, that is meiosis 2 is nothing but it is an equational division, so the chromosome number will remain constant. So the secondary spermatocytes will undergo the second meiotic division to produce four equal haploid spermatids. The spermatids are transformed into spermatozoa. Spermatids which are formed, they will get transformed into spermatozoa, and this process is called as spermiogenesis. So transformation of spermatids into spermatozoa, the process is called as spermiogenesis. Very important question again for 1 mark. After spermiogenesis, sperm heads become embedded in the Sertoli cells. So the sperm heads are embedded in the Sertoli cells and are finally released from the seminiferous tubule by the process called spermiation. So remember, you might get confused with spermiogenesis and spermiation. Spermiation is a process where the sperms get released from the Sertoli cells. So here once again I'll explain. After spermatogenesis, sperm heads become embedded. They get embedded in the Sertoli cells and they are released from the seminiferous tubule by the process. So they get released from the seminiferous tubule by the process called as spermiation. Again important question for 1 mark. Spermatogenesis starts at the age of puberty due to significant increase in secretion of gonadotropin releasing hormone. Remember, it is an hypothalamic hormone. This hormone is responsible, or it is the spermatogenesis starts at the age of puberty when there is an increase in the secretion of gonadotropin releasing hormone. The increased levels of gonadotropin releasing hormone, it will then act on the anterior pituitary gland and stimulates secretion of two gonadotropins. So it is acting on the anterior pituitary gland and it stimulates the secretion of two gonadotropins, that is luteinizing hormone and follicle stimulating hormone. LH and FSH are being released and they are secreted. LH acts at the Leydig cells and stimulates the synthesis and secretion of androgens. Androgens in turn stimulate the process of spermatogenesis, whereas FSH acts on the Sertoli cells and stimulates secretion of some factors which help in the process of spermiogenesis. Remember, LH is acting on the Leydig cells and they are stimulating the synthesis and secretion of androgens, where androgens in turn will stimulate the process of spermatogenesis, whereas FSH, follicle stimulating hormone, acts on the Sertoli cells and stimulates the secretion of some factors which help in the process of spermatogenesis. So this question also mark it important. That is about the spermiogenesis we have studied. Spermiation we have seen what happens here. So this is taking place in the males. That is about the spermatogenesis. Coming to the next, we'll study about the structure of the human sperm. This question will be asked to sketch and label the diagram will be asked. So please practice the diagram of the human sperm. A mature human sperm is highly specialized sex cell measuring about 52 to 62 micron in length. It is differentiated into three distinct regions, namely anterior head, median, middle piece and posterior tail. So it is a microscopic structure. It is divided into three parts: head, okay, and then it is having middle piece and the posterior tail. A plasma membrane envelops the whole body of sperm. So the entire body of the sperm is covered by the plasma membrane. The sperm head contains an elongated haploid nucleus. It is having an haploid nucleus. The head region is having an haploid nucleus. The anterior portion of which is covered by a cap-like structure called as acrosome. So you can see here it is having a cap-like structure called acrosome, and this acrosome is filled with enzymes that help fertilization of the ovum. So this is important for the fertilization of the ovum. So acrosome is filled with the enzyme. The middle piece possesses numerous mitochondria. The middle piece is consisting of mitochondria which produce energy for the movement of tail that facilitate sperm motility essential for sperm, that is fertilization. So for fertilization the motility of the sperm is required, and this is achieved by the mitochondria which are present in the middle piece. The human male ejaculates about 200 to 300 million sperms during a coitus, of which for normal fertility at least 60% sperms must have normal shape and size, and at least 40% of them must show vigorous motility. So they should show vigorous motility to facilitate the process of fertilization. Sperms released from the seminiferous tubules are transported by the accessory ducts, secretions of epididymis, vas deferens, seminal vesicle and prostate gland are essential for maturation and motility of sperms. The seminal plasma along with the sperms constitute the semen. The functions of male sex accessory ducts and glands are maintained by testicular hormones, that is androgens. So this is the structure of the sperm, where the sperm is divided into three parts: head, middle piece and the tail. Entire sperm is lined by plasma membrane. Head region is having nucleus containing the chromosome material. In its anterior part there is a structure called as acrosome, which is filled with the enzyme which helps in the fertilization of the ovum, whereas middle piece, even it is having a small neck there, you can see. So head and the middle piece are connected by the narrow neck. Middle piece is having numerous mitochondria which is responsible for which is providing the energy, or which provides energy for the sperm motility. It gives motility to the sperm, and the last end part is the tail. So this is the structure of the human sperm. Very important question in the exam. Diagram may be asked to be, that is draw and label. The diagram will be asked. So please practice this diagram, which is very important from the exam point of view. So this is about the structure of human sperm. Again here sectional view of seminiferous tubule is shown. Now this diagram is again asked in the exam. Please practice this diagram also. And this is also very important, which is a schematic representation of spermatogenesis, a very important question for 5 marks. We have already studied about this how the spermatozoa are being formed. So there are spermatogonia which will undergo mitosis, forming primary spermatocytes. These primary spermatocytes undergo first meiotic division to produce secondary spermatocytes, which undergo the second meiotic division to produce spermatids, which undergo differentiation to form spermatozoa. Differentiation in the sense, now these spermatids are not having the tails. You can just see here they are non-motile. They don't have the tail. So they get differentiated to form spermatozoa. So this is the schematic representation of spermatogenesis. where firstly it is undergoing multiplication phase, growth phase, and then it is undergoing the maturation phase. So three phases it is undergoing. First is multiplication phase, the second is growth phase, and the third is the maturation phase, which results in the formation of sperms or the spermatozoa. So important diagrammatic representation will be asked in the exam, so please practice this schematic representation also, which will be asked for 5 marks along with the explanation. Now this is about the, one second, we will first study about the oogenesis and then we'll study about its schematic representation. The process of formation of mature female gamete is called oogenesis, which is markedly different from spermatogenesis. So oogenesis is slightly different from spermatogenesis. Oogenesis is initiated during the embryonic development stage when a couple of million gamete mother cells are formed within each fetal ovary. So millions of oogonia are being formed during the embryonic development. No more oogonia are formed and added after the birth. Once the birth is been taken, no more oogonia are being formed. Before that only millions of oogonia are being formed. These cells start division and enter into prophase 1 of meiotic division and get temporarily arrested at this stage called as primary oocytes. So oogonia undergoes, they start dividing, they will enter at the prophase 1 stage of meiotic division and they get arrested at this stage, which are called as now primary oocytes. Each primary oocytes then gets surrounded by a layer of granulosa cells and is called as a primary follicle. So here you can see the primary follicle has been formed, which is surrounded by the single layer of granulosa cells. A large number of these follicles degenerate during the phase from birth to puberty. From birth to puberty, many of them follicles get degenerated. Now therefore at puberty only 60,000 to 80,000 primary follicles are left in each ovary. The primary follicles get surrounded by more layers of granulosa cells and a new theca called secondary. Now they are called as secondary follicles. So here one more layer gets surrounded around the primary follicles and they get transformed into secondary follicles. You can see here primary follicles getting transformed into the, they get developed and forms the secondary follicles. The secondary follicle soon transforms into tertiary follicle. Now you can see here primary follicles get transformed into secondary follicle, secondary will get transformed into tertiary follicle, which is characterized by a fluid filled cavity called antrum. So the secondary follicle transforms into tertiary follicle and this tertiary follicle is characterized by it is having a fluid filled cavity called antrum. The theca layer is organized into an inner theca interna and outer theca externa. So it is having an outer layer called as theca externa, inner layer called as theca interna. The primary oocyte within the tertiary, now the primary oocyte is present within the tertiary follicle, grows in size and completes its first meiotic division. So the primary oocyte which is present in the tertiary follicle, it is growing in size and it will complete its first meiotic division. It is unequal division resulting in the formation of a large haploid secondary oocyte and a tiny first polar body. So here the first polar body is formed by this division and secondary oocyte has been formed. The secondary oocyte retains bulk of nutrient rich cytoplasm of primary oocyte. The tertiary follicle further changes into mature follicle or Graafian follicle. It is called as very important Graafian follicle. The secondary oocyte forms a new membrane called zona pellucida surrounding it. The Graafian follicle now ruptures to release the secondary oocyte or the ovum from the ovary by the process of ovulation. Again important question for 1 mark. What is ovulation? It is nothing but here in the Graafian follicle, now the Graafian follicle gets ruptured and it releases the ovum. So this release of ovum from the Graafian follicle is called as, the process is called as ovulation. So this is the process of oogenesis, and earlier we have studied about the spermatogenesis, how the sperms are being formed. Here we have studied how the ovum is being formed. So to begin with, so we have studied about that oogenesis is nothing but here oogenesis is initiated during the embryonic developmental stage where millions of oogonia are being formed. So oogonia are formed when they are formed before the birth. So after the birth, the no more oogonia are being formed. Now the cells start, after the birth the cells start undergoing division. They will enter prophase 1, okay, and they get temporarily arrested at this stage, and it is called as primary oocyte. Now this primary oocyte will get surrounded by layer, and it is called as granulosa cells are being covered by the primary oocyte. Now it is called as, so it is called as primary follicle. So primary follicle is having primary oocyte inside. Now this primary follicle will get transformed into secondary follicle. Secondary follicle will get transformed into tertiary follicle, and the tertiary follicle is characterized by a fluid filled cavity called as, it is having a fluid filled cavity called as antrum. Okay, now this is nothing but, now you know primary oocyte is already present inside. Now that primary oocyte within the tertiary follicle, it will grow in the size, it will complete its first meiotic division, it forms secondary oocyte and the tiny first polar body. So this way the division is taking place, and the secondary oocyte retains bulk of nutrient rich cytoplasm of the primary oocyte. The tertiary follicle further changes into mature follicle or Graafian follicle. Now this Graafian follicle will rupture and releases the secondary oocyte or the ovum. In this way the ovum is being formed, and the process is called as ovulation. So that is about the ovulation. So here TS of ovary is been shown, which is showing you about the primary follicle, developing follicles. Okay, this is the mature Graafian follicle rupturing, liberating the ovum. So this is about the TS of ovary. Coming to the schematic representation of oogenesis, very important question again for 5 marks. Schematic representation along with the explanation will be asked. So here also it is undergoing 3 stages or 3 phases. It is undergoing, the first phase is multiplication phase, the second is growth phase, and the third one is a maturation phase. So here you can see primary oocytes are being formed, oogonia are undergoing mitotic differentiation forming primary oocyte. These primary oocytes will undergo first meiotic division, okay, and we have already seen that when it undergoes meiotic division, what it is undergoing, it is forming the secondary oocyte. First polar body is been formed. This secondary oocyte will again divide to form second polar body, and the ovum is being formed. So this is about the schematic representation of oogenesis. Very important question again for 5 marks. So practice both schematic representation of spermatogenesis, schematic representation of oogenesis, and there are many 1 mark questions being asked from this part of the chapter, like spermiation, spermiogenesis. Okay, so mature sperm diagram may be asked for 5 marks, and schematic representation will be asked for 5 marks. So that is about the spermatogenesis and oogenesis. So today we have completed the gametogenesis. We will stop here, and in the next class we will begin with the next part of the chapter. Thank you.